Hello, welcome to Infectious Disease, Chapter 7. In this chapter, we're going to talk about bacteria in general. One of the first things that you'll notice when you look at bacteria under a microscope is that they come in many different shapes and arrangements. This is what's referred to as bacterial morphology. A very common bacterial shape is a rod or oval shape. Sometimes these can be as long as threads. This is what's referred to as the bacillus. Most bacillus-shaped bacteria don't come in any specific arrangement, although sometimes you can find them organized into chains. The more common shape that's organized into arrangements are referred to as coccus shapes. Plural is cocci. Coccus-shaped bacteria are spherical and are in fact essentially perfect balls under the microscope. Very rarely are they found by themselves. They're most often found in different arrangements, such as in pairs for diplococci, chains for streptococci, groups of 4, in which case they'd be found as tetrads, as cubes in either packages of 8 or 64. And in this case, these are referred to as sarcina. Or finally, as clusters. And these almost kind of look like clusters of grapes under the microscope. These are referred to as staphylococci. A somewhat famous lab manual in microbiology classes in college has the title Grapes of Staph, and that's how I differentiate between streptococci, which are cocci in long chains, versus streptococci, which are cocci in clusters or like grape clusters. There's also bacteria that can form spirals. The smallest of these are the Vibrio, and these are comma-shaped or curved rods. Sometimes the Vibrio are considered to be a type of bacillus because they're essentially a rod with a curve to them. Shorter spirals are spirilla. These usually have about 2 or 3 twists to them. They often will have flagella sticking out from either end, as we'll talk about a little bit later, and can swim in sort of a corkscrew-like fashion, almost like a corkscrew in a wine cork. Longer spirals are referred to as spirochetes. And I remember the difference between spirella and spirochete because spirochetes have longer names, so you have spirochete as a longer name, than spirilla. And the spirochetes have about a dozen or so twists or coils to them. These look really kind of awkward under the microscope if you watch them swim. They bend and flex as a result of their internal or periplasmic flagella. There's some unusual other bacterial shapes that we may come across eventually at some point, but we're going to primarily forget these for the moment. Now, as we're going over through some of these shapes, you may think to yourself, I feel like I've heard of some of these words before. Perhaps bacillus, but probably more so with the streptococcus or staphylococcus, as in a child that complains about how they have strep throat, or if you've heard of anybody infected with staph. That's because there are some bacterial names that have these various shape morphologies as their genus names. This is going to transition us into a discussion about bacterial nomenclature, or how we name bacteria. For lots of different animals on Earth, it's really common for people to use common names, such as dog, cat, giraffe, elephant, and so forth. And that's because you're familiar with seeing these animals with the naked eye, and so people have been giving them common names for thousands of years. And because these names have existed for so long, they're going to be different in different languages as well. So those names in French may differ from the same animal names in Mandarin. However, in science, we would like to keep things consistent so that we can talk about organisms equivalently between different cultures and languages and through time. And for that reason, scientists have come up with what's referred to as the binomial nomenclature system. The binomial nomenclature system uses scientific names in order to name living things. And these have been used for organisms such as dogs, cats, giraffes, elephants, and other things. But they've also been applied to microbes. Now, since microbes are only really visible to scientists that study them with microscopes, nearly all microbes do not have common names. They only go by their binomial nomenclature name. The binomial nomenclature name uses two different names, genus and species. These come out of the field of taxonomy, wherein biologists are classifying organisms into smaller and smaller categories. Genus and species are the two most common categories. Whenever we use the binomial nomenclature system, we're always going to put the two names in italics, and we're going to capitalize the genus name, but not the species name. Some of these genus names come from the cell shapes. So, as we've learned about in the past, there's the Bacillus genus with things like Bacillus anthracis, which causes anthrax. There's also Streptococcus and Staphylococcus as two other genera. However, just because an organism does not have the name as the genus doesn't mean that it cannot have the shape. So for instance, E. coli is bacillus shaped, even though its genus is Escherichia. Escherichia was named after the scientist that discovered it. Let's talk a little bit about the anatomy of bacterial cells next. We get to learn a little bit more about their structures. Going from the outside in, the envelope of the bacterial cell includes the outer covering, which is the capsule, the cell wall, and the cell membrane. These are three very distinct structures that are tightly woven on top of each other and often act as an individual unit. Hence, why we refer to it as the cell envelope. All bacterial cells have the cell membrane, or a plasma membrane. That is the differentiating factor between the inside versus outside of the cell. Nearly all bacterial cells have a cell wall. There's one exception to this, the mycoplasmas, that we'll be mentioning a few times in this chapter. There are some bacterial cells, as we'll talk about, that have a second membrane called the outer membrane. Finally, there's the coating, the surface coating that surrounds the cell outside of that. Nearly all bacterial cells have some form of this, though they can be a capsule or a slime layer or different structures. Let's talk about these different structures going outside to in. So starting from the outside, going inward, the first thing that you would encounter would be the capsule or slime layer. The capsule or slime layer can be washed off of the bacterial cell and the bacterial cell would still remain alive. What the function of the capsule or slime layer is multiple-fold. So some capsules or slime layers assist with virulence, or the capacity to cause disease. For instance, this capsule or slime layer can make the bacterial cell itself really sticky and stick to host cell tissues, allowing the bacterial cell to invade the body much more easily. Alternatively, other capsules can prevent against dehydration, keeping the cell moist inside, retaining the liquid water, allowing the bacterial cell to survive for longer periods outside of the human body. Finally, another function of the capsular slime layer is in anti-phagocytosis. Essentially, your white blood cells can engulf bacterial cells as a form of host defense. In order to do this, those white blood cells need to grab the bacterial cells before they can engulf them. The capsule can make the bacterial cell so slippery that it doesn't allow the white blood cell to grab onto it, and the white blood cell can escape. This increases the chance of that bacterial cell to cause disease even more. Then, let's move from the capsular slime layer to the cell wall. As mentioned, nearly all bacterial cells have a cell wall, with the exception of the mycoplasmas. Cell walls are made of peptidoglycan in bacteria. The function of the cell wall is to keep the cell's shape and keep the cell intact, even when the cell is taking on water. Without a cell wall, the cell could take on so much water that the cell could explode or lyse. This would eventually kill the cell. There are, in fact, antibiotics that target the cell wall for destruction, including penicillin. Penicillin doesn't have any effect on human cells because we don't have cell walls on our cells. And in fact, it's fairly easy for our cells to explode if they take on too much water. Cell walls can differ in their thickness. Some bacterial cells have really thick cell walls, and some of them have fairly thin ones. The difference between the cell wall thickness can be distinguished and identified using a differential staining procedure known as the Gram stain. The Gram stain is named after Hans Christian Gram, a scientist who lived at the turn of the 20th century. He was able to develop a staining procedure that differentiated between two main cell envelope types: the gram-positive cells and the gram-negative cells. The gram-positive cells are cells that, after the Gram staining procedure, stain purple because they're able to retain the first stain used in the Gram stain, known as crystal violet. The gram-negative cells are not able to retain that initial stain, and thus are stained red or pink from the second stain used, safranin. Let's briefly cover these different steps of the Gram stain before we get into the cell envelope differences. So crystal violet is used initially to stain all the cells purple. Iodine is then used, and it acts as a mordant. Mordants clump or adhere the clusters of dye or stain to each other and to the cell itself. The third step is the decolorizing step, and in the decolorizing step you can use alcohols or acetone, essentially nail polish remover. What this step will do is remove the stain, the crystal violet dye, from the gram-negative bacteria and not from the gram-positive. It's thought that the reason for this is that the gram-positive cells have a thick cell wall and so do not allow for the easy removal of the crystal violet from the cells themselves, whereas the gram-negative cells with their thin cell wall can release those crystal violets stain easily. At this point, you would only be able to see the gram-positive cells because the gram-negative cells would be clear or transparent. So instead, we use a second stain that's of a different color, safranin. Safranin would stain the cells that have no stain, a pink or red color, whereas the gram-positive cells will remain that dark purple that the crystal violet initially caused. Gram-positive cells have a very thick cell wall and a cell or plasma membrane, but the gram-negative cells have a very thin cell wall. The thin cell wall makes them more susceptible to lysis, but they have an outer membrane in addition to their plasma membrane. The outer membrane is outside of the cell wall. We're going to talk about the function of the membrane in the next section, and in there you'll understand what advantage the gram-negative cell has over the gram-positive one.